Imagine a newborn baby, right? Taking its very first breath.
Oh, it's such a profound moment.
It is, but I mean, if you look at it strictly through the lens of biology, it is completely terrifying.
Absolutely terrifying.
Because for roughly 9 months, that baby has been developing inside this highly protected, essentially sterile isolation chamber in the womb.
Right.
And then, in just a matter of seconds, they're thrust into our world, which is, just a total microbial soup.
It's arguably the most abrupt, extreme environmental transition a human being will ever have to survive.
Yeah.
I mean, you are going from literally zero microbial exposure to complete saturation in an instant.
Airborne particles, fungi, thousands of strains of foreign bacteria. Which brings us to what I think is one of the greatest biological mysteries out there for you listening.
How does the immune system not just, completely freak out?
Exactly. Like, if an adult was suddenly exposed to thousands of novel antigens all at once, our immune system would launch a massive, fiery, and probably fatal inflammatory attack.
Good old systemic shock. But a newborn doesn't do that.
Right, and the question is why? Well, welcome to this deep dive. Today we are exploring a truly groundbreaking 2026 study published in the journal Cell Metabolism by Zheng and colleagues.
It's a phenomenal piece of biological detective work.
It really is. We are going to unpack this massive data set. We're talking over 16,000 human samples, germ-free mouse models, single-cell sequencing.
The sheer scale of the evidence is staggering.
Yeah, and the mission here is to uncover this hidden chemical engine that essentially programs a newborn's immune system. And the wildest part, they solved this mystery by looking at a baby's very first dirty diaper.
Which is just, who would have thought? But what this research reveals is that early human life operates on a fundamentally different chemical software program than adult life.
It really forces us to rethink everything. So let's just jump right in. Let's unpack the prime suspect here, which is bile acids.
Right. And when most people hear bile acids, they immediately think of, digestion.
Yeah, like biological dish soap, the stuff your gallbladder pumps out to break down the fat from a slice of pizza.
Exactly. But that textbook understanding is actually incredibly outdated. Bile acids are so much more than just digestive detergents.
They're messengers.
Very potent chemical messengers. They travel through the bloodstream, they lock into cellular receptors, and they directly communicate with our immune cells.
Okay, so historically, the medical consensus has firmly held that humans basically rely on 2 primary types of these bile acids.
Yes, cholic acid, or CA, and chenodeoxycholic acid, or CDCA.
Right, so that's the adult baseline, 2 main types. But the researchers in this study decided to analyze meconium.
The day one diaper.
Exactly. For anyone unfamiliar, meconium is that incredibly sticky, dark green first stool a baby passes on day 1. And the total volume of bile acids they found was just through the roof.
Over 9,500 micromoles per kilogram, a massive spike.
Massive. But here's the major plot twist. Over 50% of those bile acids are not the 2 standard adult types.
No, they aren't.
Specifically, 51.03% belong to this rare, obscure class called hyocholic acids, or HCAs.
And it's important to note, it's not just sitting in the gut.
Oh, right.
If you look at the newborn's blood serum on their 2nd or 3rd day of life, HCAs still make up almost 14% of all the bile acids circulating in their veins.
Which is huge. And to really grasp why that's so shocking for you listening, we have to look at what happens as we age.
Right, because the researchers track these chemical levels in cohorts ranging all the way up to people over 100 years old.
Yeah, and after the first year of life, HCAs just absolutely plummet. During years 2 to 10, they drop down to around 1.3%.
And by the time you reach adulthood, they are virtually ghosts in your system.
Yeah, they stabilize at a tiny fraction, like 0.38% in feces and maybe 2.68% in the serum.
So if you are listening to this right now as an adult, you have almost 0 HCA in your body.
But on the exact day you were born, you were absolutely swimming in it. So my immediate thought when I read this was, okay, where is it coming from?
It's a very natural question.
Right. Is the mother pumping it into the baby through the placenta before birth to, prepare them?
That was the most logical initial hypothesis. But when the research team tested the mother's blood across all 3 trimesters of pregnancy, they found no spikes in HCA.
Wow, so the mother isn't supplying a drop of it.
Not a drop. The baby is manufacturing it entirely on its own, using its fetal liver.
Wait, really? So the baby's liver is doing something an adult liver literally can't do?
Well, it's doing something an adult liver stops doing. The fetal liver utilizes a highly specialized temporary enzyme called CYP3A7.
CYP3A7.
Yeah, it's like this high-performance limited edition factory machine that only runs during early life.
Okay.
And this fetal enzyme performs a very specific chemical reaction called 6-alpha hydroxylation.
6-alpha hydroxylation. Let's break that down for the listener.
Imagine taking a standard bile acid molecule, specifically CDCA,
Right,
and just snapping on a highly specific molecular handle at the 6th carbon position. That new handle completely changes the shape and the function of the molecule, transforming it from CDCA into HCA.
And the sheer horsepower of this fetal CYP3A7 enzyme is just wild. The data shows its catalytic activity is 14.4 times higher than the adult equivalent enzyme, which is CYP3A4.
It's just churning out HCA at lightning speed.
14 times faster. That's incredible.
And what's fascinating here is that this completely rewrites textbook biology.
Yeah, because for decades, HCAs were brushed off as just random metabolic byproducts.
Exactly. But because of this dedicated high-speed fetal pathway, we now have to classify HCAs as a functionally distinct third class of primary human bile acids.
Explicitly designed by evolution to run early life.
Okay, so we're about 5 minutes in. Let's do a quick recap and bullet summary for the audience to lock this in.
Good idea.
So bullet 1, bile acids act as crucial immune and metabolic messengers, not just digestive soaps.
Bullet 2. HCAs dominate newborn biology, making up over 50% of meconium bile acids, but they drop to under 1% in adults. And bullet 3. The fetal liver enzyme CYP3A7 is uniquely responsible for this massive HCA production. So I have a question for you listening. Have you ever considered that a newborn's biology isn't just a miniaturized version of yours, but is actually running on a completely different set of chemical engines?
It's a paradigm shift,
Wow.
which naturally leads us to the most critical question of all, why?
Right. Biology doesn't expend that kind of massive energy without a profound reason.
Exactly. Why does a baby's body need to flood its entire system with this temporary chemical?
And here's where it gets really interesting. This brings us back to that terrifying first breath.
The microbial soup.
Let's zoom in on a microscopic layer of tissue in the baby's gut called the lamina propria.
Ah, yes, the lamina propria.
Think of this as the frontline battleground, where the baby's internal immune system physically meets the outside world. And the foot soldiers in this battle are called naive CD4 positive T cells.
I like to describe naive T cells as undecided voters.
Undecided voters, that's a great analogy.
Yeah, they're completely uncommitted immune cells. They just float around waiting for chemical signals to tell them what kind of specialized task force they need to join.
Right, and in the gut, they generally face a choice between two opposing destinies.
On one side, you have Th17 cells.
Key attack dogs.
Exactly. They are highly pro-inflammatory. Their job is to aggressively hunt down and fight off fungal and bacterial infections.
But they're dangerous, right? If they become overactive, they cause severe tissue damage.
Very dangerous. Now, on the other side, you have regulatory T cells, or Tregs.
The peacekeepers.
Yes. Their entire job is to be anti-inflammatory, to calm things down, and to create immune tolerance.
So a really easy mnemonic for the listener to remember this dynamic is Treg brings the peace, which is tolerance, and Th17 brings the heat, which is inflammation.
Treg brings the peace, Th17 brings the heat. Perfect. And in a newborn, you desperately need peace.
Because if those Th17 attack dogs dominate the gut on day one, the immune system will literally destroy the baby's own digestive tract trying to fight off perfectly normal bacteria.
Exactly. So the researchers obviously wanted to see what happens when these naive T cells encounter HCA.
Right, the in vitro data. They took spleen cells from mice, put them in a culture, and the results were just completely unambiguous.
When they introduced HCA, and its derivative THCA, it physically forced those undecided naive T cells to become peacekeeping Tregs.
And at the exact same time, it strictly suppressed them from turning into inflammatory Th17 cells.
It acts as an absolute biological override.
Yeah.
And if we connect this to the bigger picture, what's crucial is that the classic adult bile acids CA and CDCA could not do this.
Wait, really? Only HCA can do it?
Only HCA has this specific superpower. In fact, standard adult bile acids sometimes hindered the peacekeepers.
Wow. And the researchers even mapped this out visually using single-cell RNA sequencing and flow cytometry, right?
They did. They use a combination of algorithms to track the cellular development over time.
Okay, I read the methodology section on those algorithms, things like RNA velocity and pseudotime trajectories, and it gets incredibly dense.
It's very heavy math.
how did they actually measure a cell like changing its mind in real time?
Think of it this way. Imagine tracking a teenager who is trying to decide what to major in at college.
Okay.
Looking at a cell's standard gene expression over time is like looking at a time-lapse photo album of that teenager's changing interests. You can see the general trend.
Sure.
But RNA velocity is like sneaking a peek at the teenager's diary to see what they're secretly planning to do tomorrow.
Well, wow.
It looks at newly forming, unspliced genetic instructions to predict the cell's immediate future.
That's fascinating.
And when researchers applied these models, they literally watched the developmental path of the cells aggressively bend. Under the influence of HCA, the cells forcefully curved away from an inflammatory destiny and locked straight onto an anti-inflammatory destiny.
They watched the cellular diary change in real time. That is incredible, and it perfectly explains how babies survive that abrupt transition into the world.
Yes, without this massive wave of HCA flooding the system to act as a chemical pacifier, the baby's immune system would view beneficial breast milk and normal gut bacteria as deadly threats.
It would launch a fiery, fatal attack against its newly colonizing gut microbiome.
So HCA acts as an immunological buffer. It enforces peace in the gut.
Okay, time for another quick 5-minute recap and bullet summary.
Let's do it.
Bullet 1. Naive CD4-positive T cells must choose between becoming inflammatory Th17 cells or tolerant Treg cells.
Bullet 2. HCAs specifically drive T cells to become Tregs and strictly suppress Th17 differentiation.
And bullet 3. This creates a transient window of immune tolerance, preventing the baby's gut from attacking good bacteria and food.
So think about your own immune system for a moment. When was the last time you experienced severe inflammation?
Probably a bad cut or a stomach bug.
Right. Can you imagine your body possessing a chemical that actively forces your immune system to stand down and welcome in foreign microbes?
It goes against everything we think we know about adult immunity. Exactly. So what does this all mean for a baby's actual day-to-day health? Let's look at the graded evidence, from mouse models to human babies.
Because immune tolerance is really only half the story. The ultimate goal of this temporary peace treaty is to build a permanent, healthy microbial neighborhood.
Right, the microbiome assembly. Let's detail how HCA's peacekeeping allows this healthy neighborhood to be built. High HCA levels lead to high microbial alpha diversity.
To visualize this, think of HCA as a bouncer at an exclusive nightclub.
A bouncer.
In the germ-free mouse model, researchers gave the mice a fecal microbiota transplant, or FMT. Mice pre-treated with HCA had significantly better microbial diversity.
Because the bouncer let in all the VIPs.
Exactly.
Yeah.
High HCA promoted highly beneficial bacteria like Bifidobacterium, Akkermansia, and Fecalibacterium.
Those are the VIPs that produce healthy metabolites and keep the gut strong.
But when HCA is low or absent,
The bouncer is gone.
and opportunistic pathogens, the troublemakers take over. Bacteria like Enterococcus, Salmonella, and Enterobacter.
So it literally shapes the entire bacterial landscape. But they didn't stop there. They tested HCA against a clinical disease model, specifically necrotizing enterocolitis, or NEC.
NEC is a devastating, highly inflammatory gut disease that primarily strikes premature babies in the ICU.
And to model this in mice, they used an intense stress protocol.
Very intense. They separated neonatal pups from their mothers, fed them a harsh artificial formula, and subjected them to alternating cycles of hypoxia, which is low oxygen and cold temperatures,
Wow.
alongside doses of LPS toxins.
Wait, they hit these premature mice with hypoxia, cold stress, and toxins all at once. That sounds brutal.
They needed to trigger a catastrophic inflammatory cascade mirroring severe human NEC, and this brutal protocol caused a 50% mortality rate. The intestinal tissue was literally destroying itself.
But then they brought in the HCA. They pre-treated pups with just one single oral dose of HCA before starting the stress protocol.
And the result was stunning.
The mortality rate plummeted from 50% down to 25%, cut in half by one dose.
And the physical preservation of the tissue was remarkable. They had significantly longer intestines, less tissue necrosis.
Yeah, and at a molecular level, the HCA upregulated genes like MOOS2 and OCLIN.
Right. MOOC22 for protective gut mucus, and OCLIN for the tight junction proteins that keep the gut mucosal barrier properly sealed.
While simultaneously suppressing inflammatory markers like IL-6 and TNFA.
All the classic alarm bells were silenced.
That is massive clinical protection. But, you know, what works in a mouse doesn't always translate to humans.
True.
So walk us through the human clinical cohorts.
The data here is exceptionally robust. Researchers tracked a discovery cohort of 101 infants and a validation cohort of 95 infants, for a full year.
So nearly 200 babies in total.
Yes. They measured the HCA percentage in their day 1 meconium and then just tracked their health records at 6, 9, and 12 months.
And the results. Babies with naturally high day 1 HCA levels had a significantly lower cumulative risk of developing a whole host of issues over the next 12 months.
Dramatically less diarrhea, fewer cases of gastroenteritis, bronchopneumonia, fever, and even food allergies.
Yeah, the predictive ROC curves showed AUC values greater than 0.80 for several of these conditions.
Which means the predictive power was phenomenal. The precise chemical makeup of that first diaper strongly predicts their health for an entire year.
Mind-blowing. But it brings up this programmed handover concept. How does it all end? If HCA is this magical life-saving molecule, why do we lose it as we grow?
This raises an important question. How does the body know when to turn off the HCA engine?
Because you don't want a suppressed immune system your whole life, right? Eventually, you need your Th17 attack dogs to fight off real diseases.
Exactly. And the signal actually comes from the microbiome itself.
Wow.
As the good microbes settle into the gut and start thriving, they metabolize chemicals and produce secondary bile acids.
Secondary bile acids, okay.
These new microbial chemicals are reabsorbed, travel back to the liver, and signal that the gut is fully colonized.
So the bacteria literally send a text message to the liver saying, "We've moved in, the neighborhood is built."
Precisely. And in response, the liver hits the switch. It turns off the fetal CYP3A7 enzyme, halting HCA production, and turns on the adult CYP3A4 detoxification enzyme.
A brilliant evolutionary metabolic handover.
It really is a perfectly choreographed dance.
Okay, final recap before we wrap up. Bullet 1. HCA's immune tolerance allows beneficial gut microbes to safely colonize while crowding out pathogens.
Bullet 2. In severe disease models like NEC, HCA halves the mortality rate and protects intestinal tissue.
Bullet 3. In humans, high day 1 HCA strongly predicts a healthier first year of life.
And bullet 4. Once the microbiome is established, it signals the liver to switch from fetal HCA production to adult detoxification.
Amazing. Knowing that, how might this radically change the way we treat newborns in the ICU?
It fundamentally reframes early life. We viewed newborn biology as just an underdeveloped adult version. But this proves the perinatal environment has its own active, sophisticated software program.
So to bring this entire narrative together, HCA isn't just a random chemical. It is a meticulously timed, transient immunological shield. It's the physical bridge that allows a human to cross from the sterile womb into a microbe-filled world.
Without burning down their own biology in the process.
Exactly. And this leaves us with a truly provocative thought to mull over for you listening. If HCAs are this uniquely vital, are we standing on the verge of a pediatric revolution?
The clinical translational opportunities are immense.
Right. Could we soon see a rapid diaper test on day 1 that tells doctors if a newborn is HCA deficient?
And even more profoundly, as clinical trials continue, will the infant formulas of the future be supplemented with HCA derivatives?
To artificially recreate this magical window of immune peace for at-risk preemies, it makes you wonder, what other temporary fetal molecules have we completely ignored simply because they don't exist in adult biology?
We've been missing the magic right in front of us.
We really have. Thank you so much for joining us on this deep dive. Keep questioning the fascinating mechanics of human life, and we will catch you next time.